The overall goal of the following experiment is to transfect single or small groups of retinal ganglion cells using in vivo electroporation in postnatal mice to study the development of retinofugal projections. This is achieved by surgically exposing the eyeball and injecting a small volume of plasmid DNA solution directly into the retina. As a second step, electrodes are placed on the eyeball directly over the injection site, and electrical pulses are applied, which allows transfer of plasmid DNA into retinal neurons. Next, at the desired age, electroporated retinas and brains are harvested to visualize transfected retinal ganglion cells and their projections to the CNS. Results are obtained that show fluorescent labeling of both dendrites and axonal arbors of labeled retinal ganglion cells in their various subcortical target structures.
The main advantage of this technique over existing methods like in utero retinal electroporation is that this technique is less invasive and does not interfere with early axon guidance events such as crossing over at the optic chiasm. It also allows us to label a small population or single retinal ganglion cells in postnatal mice with precise spatial and temporal control.
To make electrodes for electroporation, break one prong off a pair of Dumont No. 5 forceps and then solder a wire at the base of each prong. Wrap the base and wires with insulation tape and fit a plastic spacer between the prongs to provide spring action. Once the electrodes are put together, connect the wire from one prong to the foot pedal, which is connected in series to the electrical stimulator. When the foot pedal is pressed, current flows through the electrodes. Next, connect the wire from the other prong to the electrical stimulator.